Today is September 17th, 2015. My name is Martin Frank, and I'm at Rush University to interview Dr. Robert Eisenberg. I've known Bob for approximately 35 years, having served with him on the Physiology study section, where I was the executive secretary and he was the chair of the study section. At the time of service, Bob was chair of the Department of Physiology here at Rush, a position that he assumed in 1976 and held for 38 years. Bob Eisenberg has been a member of the APS since 1969. Bob's research focus, not only on contractility and EC coupling, has been on ion channels, and during the course of his career, he has worked with many outstanding scientists. In his super short scientific biography on Bob Eisenberg's Living History page, he writes, I quote, "The ionic channel is where we still are, but gazing through the narrow hole that is proven to be rather like looking through a keyhole in a door. The closer you get to it, the further you can see, even glimpsing the horizon, or parenthetically, of knowledge. Occasionally even seeing a star or two when all else seems dark," end quote. During the course of this interview, we will try and explore some aspects of this statement. Bob, welcome to the Living History Project, and thank you for agreeing to be interviewed for this series. If you're ready, I'd like to ask you a few questions about your career.
Glad to be here.
Can you tell us about your family upbringing and how you got interested in science?
Well, there's a family story, and I think it might be true, and I have a few memories of it. I was a very sensitive little kid, and when my grandfather, who was a businessman, would read me stories, and these were the days when stories were Grimm's fairy tales, which are Grimm with a single M, I used to cry terribly, and he was very upset because he wanted his little Robbie to be happy. And he found the only things he could read me that I didn't cry about were things about factual material, engineers, engineering, and he read me about Thomas Edison and the George Washington Bridge, which I do remember clearly. And my mother said that that's how I got interested in science, that it was his reading to me of all this factual material.
Bob, you've talked about some of it, but tell us, were you into science in elementary school and high school?
Yes, I was very interested in science right from the beginning, and in fact, in second grade, we had a junior astronomy club, which was run by two famous mathematicians, Kripke's, who were a little bit older than me, and myself, run by a man named Macklin McCarty, who was involved with DNA discovery. So I was right involved in science right from the beginning, and I really knew it was what I wanted to do. Then by the time, it's almost a mental disease. I am doing today what I wanted to do when I was 17, almost exactly. Indeed, almost 15. I think there's something wrong with that.
In reading your CV, I noted your introduction to physiology came in high school, where you had the opportunity to interview John Papenheimer, a former APS president, on an American Heart-sponsored TV program in 1957. Can you tell us about the experience and its role in influencing your decision to go to Harvard as an undergrad?
Right. John was and he insisted that I call him John, was a career investigator, and in fact, I think the first one of the American Heart Association. So I was at Horace Mann School, and the school sent me down to interview him. They had previously sent me down to a show called The $64,000 Question, which was one of the most popular shows in the United States. And I was interviewed by Leonard Bernstein's sister, who was the key player in choosing the contestants. I wanted to go on it, but my father smelled a rat, and those of you old enough will know it turned out to be a fraudulent show. So anyway, they sent me down to interview John Papenheimer with about another 4 or 5 kids from other schools. We go to Studio 8H. Now, there'll probably be nobody old enough to know what that meant, but that's where Toscanini used to conduct the NBC Symphony of the Air. And all of the music stands were there. So I was enormously impressed, and they were shoved in one corner. We were put in another corner. In those days, they used something called a kinescope, which is a very primitive movie camera. Sat down next to John, we started taping, and of course, the machine broke. So we were there for about 4 or 5 hours instead of the hour and a half that we were supposed to be. And I talked, I'm sure, almost the whole time, because I talked even more then than I do now. And anyway, I had a lovely time, and he had a lovely time, evidently. So years later, when I applied to Harvard and went to Harvard, my father said, "You must look up Papenheimer." I had no contact with him in between. So I walked down the corridor of the old biolabs, opened the door. It said Dr. A.M. Papenheimer. I didn't know Papenheimer's first name. I opened the door, and there was somebody else, and that was his brother. It was a professor of microbiology at Harvard College instead of John being at the medical school. So he then phoned up John, and John said, "Oh yes, come over right away." And I then went over to the medical school, which is about 5 miles away, in a very awkward trip in Boston. And then John insisted that I take a graduate level course, which could produce major trouble 3 years later when I graduated, because the dean of Harvard College, John Monroe, hands me, I was a Summa, so he handed me the fancy certificate, and he looks at my grade point average. He says, "How did you ever get in that class? We have special rules that prohibit you from being in that class." So Papenheimer somehow or other stuck me in that class. That's where I first read Hodgkin-Huxley papers. I was all of 17 years old. And John thought that I should be able to understand them and explain them to everybody, which of course I couldn't do. But it set an impossible goal, which has been a very useful thing.
I noted that John Edsel was your tutor at Harvard, and you were in classes with many outstanding scientists. Can you tell us about your training at Harvard and the people you got to know during that time?
By far the most important scientific influence on me was John Edsel. Edsel at that time was editor of Journal of Biological Chemistry, and basically if you wanted to get promoted in using biochemical techniques, you had to publish in that journal. And he had actually, I later found out, turned down being a president or whatever it is, head of the National Academy of Sciences for political reasons. So Edsel had 6 or 7 tutees. Among them were many famous people. And he had 2 Eisenbergs. He had David Eisenberg in the National Academy and myself. We were unrelated, but we got to know each other very quickly because John Edsel couldn't remember who he lent the books to. So David and I had to sort that out. Anyway, Edsel and I hit it off, and we would meet every 2 weeks for the first year or so, but the last 2 years I was there, we met every week, and it was just the most extraordinary experience. He would give me a book, expect me to read it in a week or two, and I learned all my biology that way. No biology courses at all, but lots of chemistry, math, physics, and engineering, and a tremendously critical sense. It also meant that later, 40 years later, when I came back to work on some of the things that Edsel had worked on, I really do know what he knew because he taught it to me, and that turns out to be important historically.
After graduating from Harvard, you pursued doctoral work at University College London, working with future Nobel laureate Bernard Katz. Can you tell us about your experiences at University College and the people you interacted with during your time in London?
My most important interactions, of course, were with my thesis advisor, Paul Fatt, who was interested in studying the electrical architecture of cells and tissues, which is what I did. But I was extremely fortunate because Andrew Huxley loved mathematics, I loved mathematics, and we got to know each other and met, I'd say every 2 or 3 weeks for the 3 years I was there. Also, I had met Hodgkin previously at the MBL in Woods Hole, and he liked my noisy style, and I later found out it wasn't surprising since his wife is a New Yorker, and I think she's still alive, Marnie, although, and very, very talkative, but of quite more sophisticated style than my own. Anyway, Hodgkin would come visit me quite often and wound up as my external examiner, which is a crucial position there. So my most important interactions were with, in fact, Huxley and Hodgkin, and the very simple fact that I was given an empty room to do my work in. So I had to build my equipment and analyze it right from the beginning, which was not done very often, but very valuable.
In 1965, you returned to the U.S. to take a postdoctoral position in Dan Tossison's department at Duke, working with Paul Horowitz. Tell us about your postdoctoral experience, your research, and the people with whom you interacted.
The most important part of the postdoc experience was that Paul Horowitz ran a very loose laboratory, letting me and Peter Gage, Clay Armstrong, and so on interact very well. And Peter and I worked very hard on a way of separating the properties of the T-system of muscle, with the great help of my then wife, Brenda Russell Eisenberg, who had learned how to do electron microscopy. Of a very sophisticated and quantitative kind. So those were my most important interactions there. And Ted Johnson taught me how to write, which was very important.
You accepted an assistant professorship in physiology at UCLA in 1968, becoming a full professor in 1975. Please tell our listeners about your career at LA and the people and research that you conducted.
The work out at UCLA was focused on one major subject, which was the electrical architecture of cells and tissues. A whole variety of them, ranging skeletal muscle, cardiac muscle, lens of the eye, but also a variety of techniques that had to be put together. On the measurement side, we made what's called impedance spectroscopy, very, very wide band measurements, involved developing new circuits, which actually Erwin Nair was sitting next to me for about a month, and he took that circuit and exploited it and developed it much better than we did for low noise, and that's the circuit that made the patch clamp possible. And then we came back and made the Axopatch amplifier, which is still the premier amplifier, still the #1 seller after 30 years, and I'm very proud of that. There are very few electronic devices that are #1 for 30 years. Anyway, we learned to make these measurements, but in order to interpret them, we had to have quantitative estimates of the amount of membrane that was producing this stuff. So that required a set of techniques called stereology, and that was done by Brenda Russell Eisenberg, although there was lots of mathematical help and background, as people do, especially if they're married and have lots of kids, which we did. And then the theory was the part which is probably less known to the biologists, but absolutely crucial to have some equations to link the structure, which is units of centimeters, and the measurements, which are units of current. You can't link them without a theory. And that led me to work with Julian Cole, an extremely distinguished applied mathematician responsible for the design of airplane wings, and the B-47 and the 707 and so on. And we developed a neat method called singular perturbation theory, or rather I used the neat method so that we could interpret this stuff. And these results were then applied to a variety of tissues, I think there must be 40 papers on it.
In '76, you abandoned the warm confines of Brentwood and UCLA and became the first chair of physiology in Rush Medical College. What possessed you to leave the balmy weather of LA to come to the Midwest winters here in Chicago? What were the challenges you faced as the first chair of a new department of physiology?
Well, there's no doubt that it was a question of ego on my part and seduction on the part of Rush. Very well done. The dean who recruited me was a psychologist, and he knew how to play me beautifully. And the chairman of the search committee was a lovely English female scientist, actually from University College, and she thought that biophysics was clearly the best stuff in the world. So I was 33 when they started talking to me, and I went for the siren song. Not that I regret it at all. People at Rush wanted to have one classical academic department in a major clinical medical center, which it was then and is now. They basically said, you can have what you need except for space, but I didn't need a lot of space because electrophysiology doesn't need a lot of space. My challenge was to get first-rate people. And that turned out to be much easier than I thought, because what we simply did was to interview people, and the ones who were doing exciting science offered jobs. And I'm very proud, maybe arrogant, but very proud that Fred Cohen, a whole bunch of people, but Fred Cohen, Tom DeCourcy, Eduardo Rios have spent their whole careers here and are still here. And in their own areas, most people would say they are amongst the 2 or 3 leaders in the world.
As noted in my introduction, the research focus for much of your career has been the ion channel. In your short scientific biography, you referred to the channel as a keyhole. Tell us about your work to better understand the workings of the keyhole, the associated locks and flows through the keyhole or the channel.
Yes, well, there's good news and bad news. The good news is that channels are about flow. Things go through them, and life is about flow. If things are steady and there's no flow, there's a name for it in biology, it's called death. So that means that in order to interpret what's going on with a channel, you have to have a theory that has flow. The sad part is that essentially all of chemical thermodynamics, the chemical tradition, has been derived for systems without flow. It took me 40 years to realize that you can trivially show that the classical chemical methods actually violate the laws of electrodynamics, called Maxwell's equations or continuity of current. That doesn't sound like much to you guys, but 1% deviation in those laws produces 1,000,000 volts in a microsecond. This is something you cannot tolerate. So we had to create a whole set of theories to deal with this, because we're measuring single channels and we're measuring in great detail. That could be done following the path of the people who've designed semiconductors. Again, this sounds fancy, but the camera that's being used here, all of our computer technology depends on semiconductors and the fact that they really follow their equations, that you don't do things by trial and error. So I spent 10 years learning to do that and naming the equations and writing lots of stuff, but then realized that that wasn't good enough for us because we had to have finite size ions. In semiconductors, the things carrying current are not finite size. Putting finite size causes tremendous trouble. We learned how to deal with that in a rigorous way in 2007, but it was so rigorous that we cannot solve the equations for three-dimensional structures, for real structures. And the great breakthrough came about 3 years ago, where Jin-Liang Liu and I found out how to do that in a very neat and succinct way. And we now have a method that will fit the take the three-dimensional structure of gramocidin and predict all of its data, predict all of the known properties of calcium channels and ryanodine receptors, and that's mostly Dirk Gillespie's work. And in fact, we are now working on transporters. So the idea is to use mathematics that is always consistent with the electric field and use real biological structures and predict actual experiments.
Bob, tell us about your first scientific meeting and how it differs from today's scientific meetings.
Well, my first scientific meeting was so extraordinary that the story, it does need to be told. I'm in England for a month or two. I had not seen Hodgkin after I had come to England. And it was in a northern town in England, probably Leicester, I don't remember, a meeting of the English Physiological Society. And in those days, Hodgkin, Huxley, and Katz would sit on one side of the auditorium and think of the nastiest questions they could ask. So people were terrified. I was, of course, sitting in a different place and just sort of lollygogging around. And someone got up and gave a talk on how to gold plate a crayfish, so help me. And this is in the Physiological Society proceedings. You can look it up. And I put my hand up and I said I was just a student, but I wondered if he had thought that the shell of the crayfish might be high enough resistance to allow a large electric field that would plate the gold. I'm sorry, the author said this was proof of semiconductor properties. And I was trying to say it was just simple resistance, but very gently. So in one of the not so nice parts of English science, he gave me a tremendously arrogant, condescending put down, whereupon Hodgkin got up and said, "Answer Eisenberg's question. He has a point," which was the end of this man's career, and it was. Okay. This fellow then started stammering and so on. I didn't know what to do. Then he gave us the end of the talk, and in those days, the talks had to be voted on to be accepted. And so I had no idea what this procedure was, but the chairman of the meeting said, "Now we need a vote." And Hodgkin gets up, "I won't vote till I hear Eisenberg's opinion." So I'm 17, 18, I was 20 years old. And I very stupidly said, well, I think we should vote in favor. And the paper was accepted. I later found out from Katz that they were all very disappointed in me that the paper should clearly have been turned down. Now, as you know very well, Marty, I overlearned that lesson, and I haven't suffered fools gladly very often since then, although somehow I survived as chairman a long time, so I may have done it in a polite way.
How did your research area of expertise develop, and who were the key people who most influenced your career?
Well, there's no doubt that Hodgkin was my hero. He also taught me how not to administer things, but we won't go into that. I once had a conversation with Hodgkin where I said to him, you know, Prof, I was probably 18 at the time. You know, Prof, it looks to me as if you designed your assault on the nerve by starting with a differential equation and cutting out the terms. He looked at me and he said, Bob, you're the only one who's realized that. And this was, by the way, unusual, since most of his comments to me were quite critical, which I loved. So he was my hero because he combined physical science with fantastic biological intuition and incredible experimental focus and expertise, by the way, building all his own electronics. And I think that was crucially important. And then the other thing that one cannot exaggerate was how much Edsel taught me about evolution, and because there's a very strong evolutionary bias in my work. And finally, and this came from me, frankly, was the realization that the engineering work and the biological work were very closely related, because engineers analyze devices that have a purpose, which is an equation usually. Biology and physiology analyzes devices too. And the conjunction of the two is very powerful, and that's what I've been doing the last 20 years.
What do you think, as we look back at your 50-year career, will be considered your most significant contributions to physiology?
I think definitely the understanding of the importance of the electric field in the positive sense. The negative sense, which is unfortunate, is I have shown directly and very simply that chemical kinetic models violate continuity of current. And you can do this in 3 lines with high school algebra. In fact, actually, I learned that algebra in 8th grade. And that's extremely important. Nobody wants to make enemies. Nobody wants to say things are wrong. But if you're heading in the wrong direction, as one of the clinical deans here once said, the more energy you spend, the more trouble you're in. And those models cannot be used to make quantitative predictions. They can be used under one set of conditions, but I don't have to tell you, Marty, that experiments are done under a wide variety of conditions. So our models work over 5 orders of magnitude, 10 to the 5th concentration of calcium with one set of parameters, and that's because they are always consistent with the electric field.
What advice would you give students starting out in science today?
The most important thing by far is to go after smart people and ask them help. Biology cannot be done without physics, chemistry, math, anatomy, and so on. Our wonderful structural biologists depend entirely on X-ray crystallography, having been developed to almost a perfect science. What works always is to go to very smart people, ask questions aggressively in the most polite possible way you can. The way a Brooklyn Jew, although I was never raised in Brooklyn, but I was born there, learned by going to an Episcopalian school like Horace Mann. And that works in every area. In terms of science itself, just ask important questions and keep going. And of course, enjoy it. And for me, there's nothing in the world as much fun as going after the science as I'm going to do as long as I can. My father was appointed assistant professor at 88, so maybe I have a few years ahead of me.
Bob, thank you for participating in the Society's Living History program. I truly enjoyed the opportunity to learn about your career, and I'm hopeful that those who watch this video will appreciate the time and effort that you've put into the interview, as well as the many contributions you've made in your career. Thank you very much.